I wasn't sure if anybody would show up for this because I didn't know how much you felt the need for this, but I'm happy to be here to do it. Like I said, I'll do these kind of like I did the ones last term, where I'll just kind of go through the outline with you. If you have questions or if there's some specific thing that you want to know more about, let me know. I'll try to talk you through stuff. So we start at the top. I start at the top with just a brief discussion of respiration. I haven't really had a chance to get into the real respiratory things, and that won't happen until we've at least covered through the electron transport oxidative phosphorylation system. So it's once we've made that link, then you will start to see all the different controls on all the metabolic pathways because they all converge on electron transport and oxidative phosphorylation. So that's central to what's going on. That's actually will be the focus of the lecture on Wednesday. To get there, we have to, first of all, get some other central metabolic pathways under our belt, the number one of which is the citric acid cycle. And the citric acid cycle is critical because, as I said in class, it is there are two reasons for that. One is that the citric acid cycle is the primary means of oxidation in the cell, and it's therefore the primary energy source for the cell. And second, because the citric acid cycle is a little bit like what I described with respect to the pentose phosphate pathway, it is a sort of a convergence of a lot of pathways. So some of those I illustrated in this figure here that you can see, showing the amino acids and how their metabolism is involved. I introduced a couple of terms, one of which was anaplerotic, second of which was cataplerotic. So an anaplerotic pathway, I said in class, was one that uses intermediates of the citric acid cycle to fill something else up. So if we look on here, the anaplerotic pathways are shown in yellow because the arrows are going from the intermediates out to those amino acids, which means that, for example, you can use alpha-ketoglutarate to make any of those amino acids. And as I said, I'm not asking you to memorize which amino acids or any of that, though I do think you should know, for example, that alpha-ketoglutarate is involved in the metabolism of glutamic acid because that's simply a transamination. And oxaloacetate is an intermediate in metabolism of aspartic acid because, again, a transamination. And then last, pyruvate is involved in alanine metabolism for the same reason, a transamination. So those three are really easy in terms of seeing the anaplerotic nature of them. They're also cataplerotic, and though there isn't, this isn't shown directly here, you can actually see it from up here, how the aspartic acid is not showing up there, but it actually does come down in through here. Aspartic acid can readily go to oxaloacetate, for example, but you see here glutamic acid goes into alpha-ketoglutarate, and alpha-ketoglutarate goes out to glutamic acid. So there's a give and take that can happen with those pathways. Okay. So understanding those terms, anaplerotic, cataplerotic, are important, and they help to illustrate why the citric acid cycle is this sort of convergence of a lot of things. This simply shows the amino acids. If we looked at nucleotides, for example, we would see some of the nucleotide intermediates, metabolism intermediates, are found here in the citric acid cycle. And the fatty acids, acetyl-CoA, which of course is the starting material for the cycle that we've defined it as, comes from fatty acid oxidation. So all of the major, and of course pyruvate from glucose, you know that, so I don't need to tell you that. All of the major metabolic pathways converge on the citric acid cycle. So it's really, really central, really important pathway. It's even more central than glycolysis. Glycolysis feeds the citric acid cycle, but there's not a lot of glycolysis intermediates, at least not as many, that appear in other pathways as there are citric acid cycle intermediates. Okay, let's see, back to here. So we see, as I say, other pathway links. I've listed those there: heme metabolism, gluconeogenesis. This is glycoxylate. That's not right. Glyoxylate. Kevin learned how to spell. Cycle. and various shuttles that we'll talk about later, and enzyme regulators. So all those are important interactions for the citric acid cycle intermediates. I talked about the metabolism of pyruvate. That was a little bit of a review from last time, so I won't spend much time on it here. But suffice it to say, pyruvate can be oxidized under conditions of oxygen or conditions of no oxygen. And when it's in the latter, we call it fermentation. And fermentation in animals involves the production of lactate from pyruvate. In bacteria and yeast, it involves the production of ethanol from pyruvate. And when we have aerobic conditions, when oxygen is present, of course, when we have oxygen present, we have NAD+, which means we can oxidize, and so we oxidize pyruvate in oxygen-present conditions to acetyl-CoA. And NADH is needed for that reaction. Whereas in the fermentation conditions, we have an excess of NADH, and we're trying to make NAD, so we're actually reducing the intermediates of the pyruvate metabolism. I pointed out that acetaldehyde, for example, was the hangover material, and our body makes acetaldehyde by not this process, but actually this process, because we don't go through acetaldehyde, whereas plants and bacteria do that. When we drink beer or any alcoholic beverage, we have an abundance of this, so we're actually going back that way, and that's how we get acetaldehyde. I'm going kind of fast, aren't I? Pyruvate metabolism happens through the enzyme known as pyruvate dehydrogenase. Pyruvate dehydrogenase is different in bacteria and yeast than it is in animals, and that's why we see that acetaldehyde intermediate. And I've tried to draw that in this figure here. If you look pyruvate, I've drawn a straight line from pyruvate down to acetyl-CoA, no intermediate of acetaldehyde. So our pyruvate dehydrogenase is different than that of the bacteria and yeast. In both cases, however, going through acetaldehyde and then down, there's a decarboxylation going that way, or there's a decarboxylation going this way. If bacteria and yeast go down from acetaldehyde, I should get a pointer. If bacteria and yeast go down from acetaldehyde, as they do here, that's an oxidation, and so we see NAD going to NADH, giving us the same product as if we went this shortcut through the animals. However, this is where bacteria and yeast are different, is if there's no oxygen, then they're going to go upwards, in which case they're going to use the NADH back to make NAD. I talked about how pyruvate dehydrogenase has 3 subunits we call E1, E2, and E3, and each subunit has a reaction associated with that. And so as we look at that, we see, I'm sorry, I'm getting ahead of myself. There's the acetaldehyde reaction. Don't need to do that again. And there's the E1, E2, and E3. So there's 3 reactions in converting pyruvate down to acetyl-CoA. The first reaction is a decarboxylation, and that's true both in fact, all these are true in animals as well as bacteria and yeast. But we're only following here the oxidative pathway. Decarboxylation is the first step. You'll notice that the decarboxylation occurs before the oxidation occurs. So the decarboxylation is a non-oxidative decarboxylation. The product of that decarboxylation is a molecule of acetaldehyde that's bound to thiamine pyrophosphate. And I said that was one of the reasons we needed to have thiamine in our diet, because this pyrophosphate form binds to acetaldehyde. And in this form, what the function of the TPP, or the thiamine pyrophosphate, is to basically hold on to this guy, which is fairly reactive, and keep it from either being released, as can happen over here, or to react with the enzyme, which, of course, we don't want the intermediate reacting with the enzyme. TPP's job, then, is to transfer the acetaldehyde to a coenzyme A. So that's what's happening in this step. And it's in this step that the oxidation actually occurs. Okay? So the oxidation occurs because the acetaldehyde is converted into an acetyl group. And that conversion of the acetyl group means the electrons have to go somewhere. And the electrons actually go to this lipoamide, which contains a disulfide bond. Excuse me. So TPP is transferring and oxidizing acetaldehyde to lipoamide to make this acetyl lipoamide. So when you see this acetyl group, you know an oxidation has happened. The acetaldehyde is getting oxidized. The disulfide bond is getting reduced. Okay? So we have a reduction that's here. Now we have an acetyl group. All we have to do is excuse me, transfer the acetyl group to a coenzyme A. That's not even shown on here, but there's a coenzyme A that comes in, and we get acetyl-CoA. Next, what has to happen is that this sulfhydryl, this reduced lipoamide, has to be reoxidized. So it gives up its electrons to FAD to become FADH2. And FADH2 gives its electrons up to NAD and becomes NADH, as you can see right here. Okay? So as a consequence of that, we've had oxidation of pyruvate, we've had decarboxylation of pyruvate, and we've had production of an NADH. Okay? That's the net production of this pathway, as well as an acetyl-CoA. Okay? This starts as an FAD and becomes an FAD when it transfers electrons back over here. At this point, the enzyme is then ready to go and do another cycle of the same thing. But these are the three steps of the process. Yes, Leo. Yes. Okay. So I'm not sure if I'm answering your question correctly, but this is transferred to a CoA. This is a sulfur acetyl bond, and this is a sulfur acetyl bond. These two are at the same state, and that leaves behind a sulfhydryl instead of a disulfide bond, which means that it's got two sets of electrons, all right? Or two electrons, really. Those two electrons have to be taken away from here so we can regenerate the disulfide bond. And they get put on here. Then this transfers the electrons onto here to make this. Okay. Whenever you see a reduction like this or a reduction like this, it always involves two electrons. It's always two electrons. Electrons travel in pairs. We'll see this in the electron transport system in an interesting way in that they start out in pairs, but they end up moving one by one at the end. And it's one of the reasons that oxidative electron transport can actually give reactive oxygen species. So when you start splitting electrons, you have the potential to make reactive oxygen species. Am I answering your question? I'm getting off track there, but that's important. Yes? Yes. Uh-huh.
You're describing in the second reaction, use the one oxidizing, or is it
Okay.
enzyme? I thought they were like...
Okay. I used bad words there. I used the wrong words there.
Okay.
So really all that they are handles. That's really all that they are. Oxidations are happening as things are passing from one to the other. And I did say TPP did the oxidation, but it doesn't. Okay. So it's enabling the oxidation, but the actual oxidation, the actual catalysis is occurring by these subunits. So thank you for making me make that clear, because I don't want people to have the wrong impression. Okay? Okay. All right. So that's what's happening in the pyruvate dehydrogenase reaction. There's mechanism, which, as I said, I'm not going to go through mechanism, but you can actually see what's happening if you look at this, if that helps you to envision things. Here is, for example, the disulfide ring, which when it gains electrons, you see a sulfhydryl, and you see the acetyl group attached to it here. You can see the CoA coming in here, and you see what's left behind, which is two sulfhydryls. Those pass electrons off to FAD. FAD passes electrons off to NAD to make this. Okay? But I don't care that you know the details of the mechanism. I want you, if you knew the figure prior to this one and you understood that, I think you're perfectly fine. Okay. Lipoamide. Lipoamide is an important nutrient. It is essentially a vitamin. Okay. Lipoic acid or lipoamide, same thing. Lipoic acid is the red component here or the blue component here. and it's attached to the side chain of a lysine in the pyruvate dehydrogenase. Okay? So when you attach it to a lysine, you get lipoamide as opposed to lipoic acid. The free form would be lipoic acid. And of course, here's the oxidized form because it's got a disulfide. Here's the reduced form because it's got 2 sulfhydryls. Okay. I said I was going to come back and talk about regulation in class. I really didn't do that much. It becomes a little bit relevant as we talk about the electron transport system. And it's also relevant as we talk a little bit about the citric acid cycle. Okay? There's really 2 things here that I would take away in terms of regulation. One is the importance of NAD. Okay? NAD, as you know, is going to be necessary for the oxidation. You don't have to look at this figure to figure or try to figure out where NAD fits in there. You know NAD is necessary. And so that involvement of NAD in any oxidation reaction is going to be critical. So that's one of the primary ways in which this enzyme can be regulated. If you don't have NAD, then nothing else is going to matter. You can have this guy in the dephosphorylated form all you want, but if there's no NAD, the enzyme isn't going to catalyze anything. Okay? Now, so the only message, so that's one message there. NAD is critical for this enzyme. The other message is that phosphorylation affects the enzyme's activity. Okay? So you phosphorylate it to inactivate the enzyme. You dephosphorylate it to activate the enzyme. Now, I find this figure, to be honest with you, confusing because it's the kinase, it's actually the regulation of the kinase that determines whether the enzyme is inactive or active. So the tendency is to say, well, look, hey, you got NAD, you're going to make this enzyme inactive, right? But that's not what it's saying. That red arrow means that it's inhibiting this, right? Presence of NAD inhibits the kinase, so the kinase can't put phosphate on there, and therefore the enzyme is left in this state. So the figure is not the best one I could have chosen to illustrate this principle. But the important thing here is really just noticing the difference between these 2 forms. The phosphorylated form is inactive, the dephosphorylated form is active. Okay? This does play into bigger pictures in terms of signaling. Okay? And I always like to just kind of do a little mental exercise with that with respect to phosphorylation. Okay? So the phosphorylation of this enzyme is going to happen when phosphorylation is going on inside of cells. And so phosphorylation is going on inside of cells when? Thinking about the signaling with the GPCR, the beta-adrenergic receptor, the glycogen, glucose metabolism, right? Okay. So what's phosphorylation going to do there? Means glucose will be released, right? Okay. So if you want glucose released, then what do you have to do to release glucose? You break down glycogen and you synthesize glucose via gluconeogenesis. Those 2 pathways are favored in liver cells. What does it take to synthesize glucose? Do you want pyruvate dehydrogenase to be active or inactive? You want it inactive because if it's active, you're going to get a futile cycle, right? And that's what you're seeing. Right? So these all are interconnected. Okay? And there's a coordinated response. That's one of the things you're going to see in respiration is this coordinated response that you don't have one part of a pathway interfering with a different part of the pathway because that would not be energetically productive. Okay? The only times you see cells that do things in futile cycles are where they're trying to make heat. That's the only time you see it. And this would not be routinely being done. Okay? So it's consistent then that when you're phosphorylating things that you want this enzyme to be inactive. Okay? Okay. There's the handle, there's the CoA, and you can see how the handle is way bigger than the thing it's carrying. Okay? Not too much of a point there, but I like to have people see it anyway. Citric acid cycle. Okay, you got 8 reactions. All right, 8 reactions. And I think I promised I was going to show you a way to memorize those, and I haven't done that. I'll do that briefly. But you don't need to memorize the structures, as I said. You need to know the names of the enzymes, you need to know the names of the intermediates, and you need to know how many carbons are in each one. Okay? Well, we start with this reaction as a starting point, 2-carbon piece of acetyl-CoA, a 4-carbon intermediate oxaloacetate. Put them together, we get a 6-carbon molecule. This reaction, I said in class, was very energetically favored because you're breaking a bond of this right here. That sulfur acetyl bond is a very high energy bond. The breaking of it releases energy, favors this reaction. Enzyme that catalyzes it is citrate synthase. Second reaction is the only reaction that's catalyzed by an enzyme whose name doesn't really tell you very much, and that's a conatase. It's named for an intermediate in the process that I haven't shown you called a conatate, but that's where the name of the enzyme comes from. And all that's happening in this reaction is you are isomerizing citrate at the top down to isocitrate at the bottom. So this OH group on position 3 is moving down here to the position 4 below. Now, the next step in the pathway is the first oxidation that happens. Isocitrate with 6 carbons gets oxidized to become alpha-ketoglutarate with 5 carbons. And you can see that this guy right here, what I call carbon number 6 on this, which was attached to carbon number 3, has disappeared. And that's the source of the CO2. This is an oxidative decarboxylation, so we have to have an electron carrier, so there's NAD becoming NADH. And the enzyme there is isocitrate dehydrogenase. As we go to the next reaction, this is the reaction catalyzed by alpha-ketoglutarate dehydrogenase. This was the enzyme that I said used the same 5 coenzymes as pyruvate dehydrogenase, very, very similar mechanism. The 5 coenzymes, I'll repeat, are FAD, NAD, coenzyme A, thiamine pyrophosphate, and lipoic acid. All 5 of those are needed, both by pyruvate dehydrogenase and by this enzyme. This is just another oxidative decarboxylation, NAD going to NADH, CO2 being released, and the product of this is succinyl-CoA. Now, alpha-ketoglutarate, I want you to remember, was an intermediate that I said was important for glutamic acid metabolism, and so wherever you see that alpha-ketoglutarate, it means it can either be taken away to make glutamate, or it can be brought in to feed the cycle. And so that's a really, really good flexible point of the cycle. Succinyl-CoA is important for metabolism of heme, and we won't talk about heme this term, but I will tell you that that is the case, that it does that. The next reaction is catalyzed by the enzyme who's named for the backwards reaction, which is succinyl-CoA synthetase. The reaction in the direction of the citric acid cycle is to take succinyl-CoA and make succinate. So the enzyme was named for going from down to up, which is also fine. This reaction is the only reaction in the cycle that is a substrate level phosphorylation. We see GDP going to GTP, as you can see here. As I said, some organisms use ADP into ATP. The energy for this comes from the fact that, again, we have this CoA attached to excuse me, a carbonyl group, which is what we have in succinyl-CoA. Breaking that bond releases energy, and that's the driving force for making the GTP. At this point, we have a symmetrical molecule, so I can't tell which end has 1, 2, 3, or 4. You can't do that. So it doesn't matter from this point forward what the numbers are on those. I showed briefly the mechanism here by which this occurs involving a phosphate, which makes a nucleophilic attack here. That attachment at this point causes the release of the coenzyme A and provides a target now for attack by the electrons actually on this nitrogen. This seeking the electrons on this nitrogen. And no, I said it the right way the first time. The electrons here are seeking this phosphate, which is slightly positively charged. The result is the release of a succinate and the attachment of the phosphate here. The phosphate covalently attached gets removed by the NDP, which in this case is GDP, to make GTP, and then we're right back where we started. Okay, I'm just sailing through these, so if you have questions, let me know. This was the first of the reactions I said was like reactions that you'll see in fatty acid metabolism. This reaction is an oxidation. It's an oxidation between two carbons joined by a single bond. We haven't seen oxidations like that to date. And those oxidations can't use NAD, they actually use FAD. And the result is, once the electrons and the protons have been taken away, a double bond in a trans configuration, as you can see here, and the production of FADH2, the enzyme succinate dehydrogenase, and this is the third reaction of the citric acid cycle. Okay. This was the only enzyme I said that was not found in the matrix of the mitochondrion. This is bound in the membrane, the intermitochondrial membrane that we'll see is so important. And I mentioned that this enzyme was also involved in the transport of electrons in the electron transport system, as we will see on Wednesday. The next enzyme in the cycle is fumarase, and this involves addition of water across a double bond, as you can see here, to produce this intermediate here, which is known as malic acid or malate, as you see here. And this this adding of water is exactly what happens in fatty acid oxidation as well. It's a common mechanism to introduce an oxygen into a molecule. And the next reaction is essentially the last reaction. It's the only one we haven't talked about so far. This is the last oxidation. We see the oxidation of malate down to oxaloacetate, the NAD going to NADH, of course. And this is the unusual oxidation that has a fairly positive delta G0 prime reaction for its catalysis. And it's catalyzed by malate dehydrogenase. And as I noted, that this reaction is actually pulled by the citrate synthase reaction. It has a very negative delta G0 prime. So as a result of the citrate synthase reaction, this guy gets gobbled up and converted into this guy gets converted into this, which then gets gobbled up and made into citrate. Any questions? Is that fast enough? There's the cycle with all the things put onto it. Yeah?
Would you like us to know at what point you're in the mitochondrial membrane or matrix? Like, I feel like
Okay.
the cytosol is a difference in glycolysis and then...
Sure. So I think, you should certainly know that this is all occurring in the mitochondrion, yes. So the matrix is where 7 of the enzymes are located, and this succinate dehydrogenase is located in the intermitochondrial membrane. And yes, I will expect you to know that because that will become important when we talk about electron transport. But all the other enzymes are in the matrix. And that isolation of these reactions in the mitochondrion turns out to be important because, as we will see, fatty acid oxidation, which involves conversion of fatty acids into acetyl-CoA, also occurs in the mitochondrial matrix. So this is convenient in the first place. So the acetyl-CoA from the fatty acid oxidation is coming in very readily there. But more importantly, fatty acid synthesis, which also requires ultimately acetyl-CoA, is not occurring there because you don't want these two things going on at the same place at the same time unless you've got regulation. Instead, fatty acid synthesis is occurring in the cytoplasm. So that location here turns out to be pretty important. Yes, sir? I'm sorry? So this, actually, I never even noticed that. You're right. That is the equivalent of FAD, yeah. So what happens with this, as we will see in electron transport, is that there's a thing called coenzyme Q that carries the electrons, and that's what this is trying to show. I didn't even realize that was on there, so yeah. Okay. So there's the other pathways. I'm not going to go through that. I've already mentioned those to you briefly. Anaplerotic, cataplerotic, I've talked about that already too, so I won't go through that. And the reversal of the pathway, which is a little unusual, as I mentioned in class. But if you reverse the pathway, which some bacteria do, they can actually use the reversal to actually take in carbon dioxide and incorporate it into bigger molecules. And so if you reverse the pathway, every time you go through, you're going to release an acetyl-CoA. So the net result of reversing the pathway would be to convert carbon dioxide into acetyl-CoA. And if you remember, and this happens in bacteria. So if you remember what I said about the glyoxylate cycle happens in bacteria. So bacteria could take carbon dioxide, make acetyl-CoA, and since they can use acetyl-CoA to make glucose, they could convert carbon dioxide into glucose with the reversal of this pathway, which is really an odd thing to do. It takes energy. It takes a fair amount of energy to run this pathway backwards. It also will take a fair amount of NADH to make this pathway run backwards as well. Okay. Okay. So, glyoxylate cycle. Briefly, glyoxylate cycle short circuits the citric acid cycle. It short circuits it specifically from this point of isocitrate, making the glyoxylate and succinate here. The short circuiting is made possible first by the enzyme isocitrate lyase, which is found only in plants and bacteria. And that produces glyoxylate, which when it's combined with an acetyl-CoA makes malate. Meanwhile, the succinate that was also produced by this splitting is converted into malate. So by this point, we have 2 malates, and then we have 2 oxaloacetates. So we started with 1, and we go all the way through the cycle, and we end up with 2, which means we're producing a net of 1 each time we turn the cycle. And that means that extra oxaloacetate can be used for something, and the something commonly used for is making glucose, because oxaloacetate is an intermediate in gluconeogenesis. And that means that plants and bacteria can convert acetyl-CoA into glucose. And we can't do that because we can't short-circuit the cycle. Yes? So that's a really good question. So that, again, comes back to this question of, well, when do they run the glyoxylate cycle, and when do they run the citric acid cycle? And the answer really is more in line with what I said in class. That is, when there's a shortage of NAD, there will be an abundance of NADH. So you're finding ways to reduce your need to have NAD, and this pathway does that. There's only 1 NAD that's required and 1 FAD that's required, whereas in the other cycle, in the citric acid cycle, you've got 3 NADs and 1 FAD. Now, to come back to your question, do they do it just to make glucose? The answer is, for the most part, yes. So they do it, and so when do we make glucose? We make glucose when our cells have energy. We make it in our liver cells, and our liver cells are usually fairly flush with energy. So they can turn on the synthesis of glucose because the energy that's needed is there. They have NADH. They have ATP that's necessary for making that. Okay. Here, these guys have to have an abundance of energy to do that. And an abundance of energy will occur when they have plenty of NADH. So that's why the answer to the question is yes, for the most part, they're going to do that. They're going to be making glucose only when running this cycle only when they need to make glucose. I'm sorry? Plants and bacteria. Yep. Mm-hmm. Okay. Oop. All right. 2 enzymes that are required for the glyoxylate cycle: isocitrate lyase. I don't know why I want to say succinate, and the other being malate synthase. Those 2 are unique to plants and bacteria, so we don't have those, and that's why we can't run the cycle. Overview of the pathway looks something like that. This gives you a flavor for all the different things that acetyl-CoA is involved in. You see it coming from fatty acids, you see it coming from sugars, and you see it coming from proteins and amino acids. You can also get some of it from breakdown of nucleotides, which aren't shown on here. But then you show that the common place where this can be oxidized, if we're oxidizing, is to go here. And then we'll see about these 2 processes starting on Wednesday. Okay. The animal's dilemma, of course, is that they can't make glucose from fat. I've made that point, hopefully emphatically. And I talked about ketone body metabolism, which is important then. Animals have a constant need for glucose. They're not constantly eating glucose. And as I said in class, animals only have about a 24-hour supply of glycogen to supply glucose for the body. So you went 24 hours without eating any glucose, you would need to have alternate sources. Some of those alternate sources will come from proteins, and you'll start actually breaking your proteins down to get amino acids to make things like oxaloacetate and so forth, or alpha-ketoglutarate from glutamate to make glucose. As you go further along that pathway, you start running out of those essential things to make glucose. Or if you have a very immediate need, okay, for glucose, before you can start breaking down proteins, you need to have a very immediate energy source. And so this is where ketone bodies come in. Ketone bodies make a 4-carbon intermediate, starting with actually 3 2-carbon intermediates. 1, 2, and then there's a third one right here. Okay? So what the body is trying to do at this point is to provide a source of acetyl groups to cells. Now, an easy way to do that is with glucose, because glucose goes to pyruvate, pyruvate goes to acetyl-CoA, and you feed the citric acid cycle. If you don't have glucose, it's nice to have an alternate way to get those 2-carbon pieces into the citric acid cycle. So that's what the ketone bodies are doing. Another reason that the body is using ketone bodies is that they're water soluble. So you can dissolve these in water. They can go in the bloodstream very readily. You don't have to package them up like you do fats and fatty acids. So consequently, these can be made and released into the bloodstream very quickly and supply the needs of the body. So that's what happens. There's this enzyme called thiolase that I said you should know, and we'll talk about thiolase later. But thiolase joins these 2 guys to make a 4-carbon intermediate. And I don't care if you know the name. The 4-carbon intermediate is converted into a 6-carbon intermediate by adding another acetyl group, and then the acetyl-CoA itself is split off to leave this 4-carbon intermediate acetoacetate. You should know this name. I don't care if you know the structure or not, but you should know the name. Acetoacetate is a ketone body. This guy is chemically unstable, as I said, so it will spontaneously decarboxylate to leave acetone. And acetone is the stuff that I said if you smell it on somebody's breath, tell them to go see a doctor, because there's a decent chance they may have diabetes, because what's happened is they've gone very hypoglycemic. The body needed a quick energy source, so it made this. And this was coming out the lungs and telling you that this person was low on energy. One of the things the body does to avoid this, because this is a non-productive way, just a second, Leah, this was a non-productive way to get things, because this isn't going to go to the cells and give 2-carbon pieces. It's going to go out the lungs, and you're going to lose it, right? So what the body does is it converts acetoacetate into beta-hydroxybutyrate. That makes a more chemically stable form. And this guy will then go to the target cells. Now, I'm not sure if I said this in class, but if I didn't, I will say it here. How do the cells get the acetyl groups? Okay? The cells get the acetyl groups by reversing the whole process. So they go up here and they say, okay, here's beta-hydroxybutyrate. We're going to make a 4-carbon intermediate. We're going to go back up here. And here's what we finally end up with. So the cells ultimately are getting these, starting with these things down here, just running it backwards. Consequently, the cells then have acetyl groups that they can feed the citric acid cycle with. Yes, ma'am? So this is a good example of the liver feeding the rest of the body. Mm-hmm. Exactly. Right. So the liver is amazing, what the liver is doing to take care of the body. Yep. That's right. Yep. Good question. So when, and your question, Leah, is also good because it reminds us, partly me and partly everybody else, to think about, there are differences between what happens inside of cells and what happens in the body. So the Cori cycle was a good example that you saw last term. We saw the Cori cycle was the liver was feeding glucose to the rest of the body, and the body was feeding lactate back to the liver to make more glucose, right? So there's a difference between what happens in the body and what happens in a cell. If we had both of those things going on inside of cells, we would have a futile cycle. making of glucose and breaking down at the same time, we'd have a futile cycle. But because it's occurring in one part of the body and helping another part of the body, we don't look at it as a futile cycle. Make sense? Yes. So breakdown of fatty acids, we haven't seen that yet. Breakdown of fatty acids will produce this. So this is a way of getting energy from fatty acids that are going then into the bloodstream to go back and feed the brain. Yep. Okay? Is acetone harmful to the body? Well, that's like I said in class. You know, I wouldn't recommend that you go out and drink a beaker of it. I don't know that it's overly toxic to the body, but also the concentrations that are there are pretty minimal. So I think you probably wouldn't want to be producing that for too much for too long. It is an organic chemical, and you know, you worry about denaturing proteins and things like that. I don't know that there's any specific problems associated with it, but again, it wouldn't surprise me. So don't go out and drink your acetone, your fingernail polish. Well, it's very volatile. It's very volatile, which is why you smell it. So it'll volatilize into the air. Yeah, sure. Yeah, acetone is a liquid at room temperature, but it's if you've ever worked with it in organic chemistry, which I'm sure you have, you know it's got a strong smell, and that's because it readily evaporates. Yeah.
So the body, or the liver rather, is mostly doing this to transport things more easily. Is that
The liver is finding a way to put stuff out in the bloodstream for the bloodstream to take that to the target cells.
because if they put acetylcholine, that would be active so the foods wouldn't work well. So transport it.
That's actually a very good question. Why doesn't the liver dump acetyl-CoA into the bloodstream? And, you know, off the top of my head, Dwight, I don't know the answer to that question. Nobody's ever asked me that question before. That's good. Think about that for a second. One of the things that I will tell you that cells avoid doing is putting free fatty acids out into the bloodstream. And part of the reason for that is that they act like detergents. And so they will go out and they hit a protein. What are they going to do when a detergent hits a protein? It's going to denature it. Now, this isn't large enough, I think, that would happen. But maybe it's the CoA part of it. That may be part of it. I don't know. But fatty acids do travel in the bloodstream, though. And the way that they travel, they either get packaged up into the lipoprotein complexes, or, and this is the more common way for fatty acids, they get gobbled up by serum albumin. Serum albumin is the most abundant protein in your blood. And it's gobbling up all those free fatty acids. It carries other things as well, but it carries them very efficiently so that they don't get exposed to go out and do other problems. Yeah, good. Okay, I'm taking long. I told Indira, I said, oh, this won't take longer than an hour. I'll probably okay. This thing I showed simply to point out that this 6-carbon intermediate, which you see in the ketone body synthesis, HMG-CoA, is an intermediate in both the synthesis of ketone bodies, which this guy right here, and in the production of cholesterol. And we'll talk about that when we talk about cholesterol metabolism. Okay, that's done. I talked about exactly one outline there. Oh boy, I've got to get moving. All right, obesity, fat mouse, skinny mouse. Okay, saturated fatty acids. All right, so we've got saturated, no double bonds. We've got unsaturated, at least one double bond. Whenever we have biologically produced fatty acids, they're almost always in the cis configuration, and that cis configuration causes a bend, as you can see. The saturated fatty acids have, you'll notice that they differ in carbons by 2, and that's because they're actually made by adding 2 carbons at a time during the fatty acid synthesis process. And the unsaturated fatty acids are shown there. One of the things this figure gives, I think, the wrong impression about it is it makes it seem as if the trans fatty acids that occur in nature are more abundant than they really are. Percentage-wise, this is really rare, this is really rare, and this is really rare. Okay, so on a percentage basis, it's extraordinarily rare that organisms produce trans fatty acids. Bovines will produce a little bit. Okay, but most of the trans fatty acids that you find in food, when you hear about trans fat and so forth, are made by chemical processes in the processing of the food. So the cis are by far the more common. I talked about 2 numbering systems. the delta numbering system, which starts at the carboxyl and moves towards the methyl, and the omega system, which starts with the methyl and goes towards the carboxyl. I pointed out that essential fatty acids can't be made in mammals past position 9. So oleic acid, which has a double bond at position 9, is non-essential. We can make that one, but we can't make linoleic or linolenic because they have double bonds past delta 9. Okay, fats and oils. I said the difference between a fat and an oil was only that a fat will be solid at room temperature and oil will be liquid at room temperature. They are both triacyl glycerols, which is what you see on the screen. And the property about whether it's fat or solid, whether it is liquid or solid at room temperature, really has to do with the length of the fatty acid and the degree of unsaturation. The more unsaturated and the shorter the fatty acids, the more likely it will be a liquid. That is, it'll have a lower melting temperature. And the longer the fatty acids and the more saturated they are, the higher the melting temperature. That will turn out to be important as we get to membranes. Okay. I talked about enzymes that break down fatty acids, and those are break down fats, and those are called lipases. We'll also see they break down the glycerol phospholipids as well. Okay. Next, I talked about the glycerol phospholipids, and these guys have a glycerol backbone, just like the fats do. They have 2 fatty acids attached instead of 3. And the third position is occupied by a phosphate. That phosphate becomes a point of attachment for other molecules like serine, like ethanolamine, like choline, et cetera, et cetera. Those compounds are called phosphatides or phosphatidyl compounds, and they're very important in membranes. There's some of the compounds that are attached. Phosphatidylcholine, shown there. In every event, the molecule that's attached at that third position is relatively polar. And so when we look at a glycerol phospholipid, we see that we have a polar end and a long nonpolar end. And those go into lipid bilayers, as I've talked about before. Okay. Let's see. Cardiolipin is an unusual one in that it's a diglycerol phospholipid. We see there's a glycerol, there's a glycerol, there's a fatty acid, fatty acid, fatty acid, fatty acid. There's a phosphate, and then in the middle, there's a third glycerol. So it's a really unusual one. These guys are found predominantly, or almost exclusively, in the inner mitochondrial membrane. As I pointed out in class, these guys play a role in the process known as apoptosis. And basically in apoptosis, there's a signal that there's been some kind of damage that's occurred. And when that damage occurs, the cytochrome C is released by these lipids in the inner mitochondrial membrane so that it can travel out into the cell. And that tells the cell we got problems, we need to commit suicide. And that's what they do. That's what apoptosis is. Okay. Sphingolipids, another class of lipids. Similar overall to the glycerol phospholipids in having 2 long tails that you see here and another end that is polar, and that's this guy up here. It's to that end that are attached either phosphate or other things like sugars. So we see, for example, that the overall sphingolipid shown here on the right looks very much like a phosphoglyceride. This is one of the sphingolipids that contains a phosphate. Most of the sphingolipids don't contain phosphate. They contain sugars. But this one contains phosphate up at the top, and it's known as sphingomyelin. Sphingomyelin, of course, is a compound that is found in the bundle sheath cells of nerve cells. They provide a sort of a protective barrier on the outside membrane of those. Again, the polarity of these, very much like the glycerol phospholipids, is not drawn as a tail, but there's a long nonpolar tail there, a long nonpolar tail there. So we have a nonpolar end, and then we have another end that's very polar, as you can see here, and that was sort of shown in the last figure as well. Okay, categories. Sphingomyelin. As I said, one of the few that actually has a phosphate. It has phosphocholine on the end of it. If we have a glycerol phospholipid that contains a sugar, if it's only a single sugar, it's known as a cerebroside. If it has a complex set of sugars, it's known as a ganglioside. Okay, and sphingomyelin there. Not sure what that is. Oh, there it is. Okay, it's just a glucose cerebroside, meaning it's a cerebroside that has a glucose on it. Eicosanoids are 20 carbon compounds that are made from arachidonic acid. The arachidonic acid contains 1, 2, 3, 4 double bonds. All those double bonds are in the cis configuration, as you can see there. We can take the same molecule and draw it like this. And I draw it like this because it looks like what become prostaglandins, which are one of the types of eicosanoids. In prostaglandins, these two things are joined into a ring structure, and an oxygen is added, and that's a reaction catalyzed by a cyclooxygenase, making a ring, the cyclo part, and the oxygenase adding the oxygens to that. Eicosanoids include things like prostanoids, which are the prostaglandins, and thromboxanes, as we'll see in the next figure. Prostaglandins, there's that ring with a 5-member ring with the 2 oxygens attached. This is one of the primary prostaglandins because this is what's made directly from arachidonic acid. It's processed into other ones like this, or it's processed into the thromboxanes, which are shown here. So the conversion of these guys into this is important in the body. Thromboxanes are involved in blood clotting. And it turns out that the cyclooxygenases that make this guy are inhibited by non-steroidal anti-inflammatory drugs known as NSAIDs. And those guys are examples are aspirin and ibuprofen, for example. And that's why we think of these NSAIDs as being painkillers. They will stop the production of prostaglandins. And they're also blood thinners because by stopping the production of prostaglandins, they're stopping the production of thromboxanes, which are involved in the clotting process. The third class of molecules here, the leukotrienes, I talked about in class as being involved in allergic reactions, inflammation, and so forth. But they're not made from these. They're actually made directly from arachidonic acid. Okay. Now I'm starting to pick up the pace a little bit. The last class of eicosanoids are the cannabinoids, an example of which is anandamide, which is an endocannabinoid, meaning it's made in our body. We have cannabinoid receptors in our brain that are binding this molecule. And as I mentioned in class, they are involved in reducing pain. They give euphoric feelings and so forth. And they also are involved in a response for eating. One of the reasons people get munchy when they get high is that they are stimulating with an artificial compound those same receptors in their brain that tell them to eat. And a third, I didn't mention this in class, but I'll mention it here, another interesting fact about this receptor, which is the one that's stimulated. These endocannabinoids are made when you run, for example, or heavily exert yourself. Another thing that binds to this receptor is chocolate. And so the euphoric people that people get with eating chocolate probably comes from stimulating that same endocannabinoid receptor that's used by this and is bound by this, which is, of course, the active ingredient in marijuana. This is one of the active ingredients in marijuana. Yeah. Marijuana actually has different types of marijuana have different mixes of different cannabinoids. Okay, this is the most common one that's there. It's probably the most abundant one that's there. But there's something like 80 different cannabinoids that are found in marijuana. So people are still trying to sort out, what the mix gives you, say, the best high or the best whatever, or maybe the worst effect or whatever associated with that. So it's an interesting story. As I think I mentioned in class, this guy was discovered first. So we knew what the active ingredient was in marijuana, but nobody had any idea why it was having this effect. They knew there had to be some receptor it's binding to, and they couldn't find it binding to any receptor. So they had to really look hard to find the receptor, and once they found the receptor, they found this. So it was an interesting story, but this was known long before the receptor itself was known. Yeah.
So are the endocannabinoids made from arachidonic acid as well?
Yes, the endocannabinoids are made from arachidonic acid and ethanolamine. That's an ethanolamine. So if you went from this guy around here, just from that point around here, that's arachidonic acid. So it's a real simple synthesis that cells, that the body does to make that.
And are there a large number of different endocannabinoids?
You know, I don't know the answer to that question. I think the answer is yes, but I don't know. This is by far the most common one that people study. But I think the answer is yes. And there are different, the abundance, this is another thing that was surprising. The endocannabinoids are, the endocannabinoid receptor is the most, if I recall this correctly, is the most abundant G protein coupled receptor in the brain, which means it's everywhere. And why they had the difficulty identifying in the first place, I don't know. Yeah, kind of cool. Okay, let's see. I'm trying to get through this, so let's see, guys. And I'm trying to stop you. You're always welcome to ask questions, of course. Cholesterol, know the rings, right? Don't care about the numbering or any of that, but know the rings. Know that cholesterol is, and I didn't mention this in class, but I'll just say it here, cholesterol abundant in animals. Plants don't really hardly have any cholesterol, but they have related compounds, as you can see from the figure there, very related compounds. Cholesterol, I thought I fixed that. Damn it. This thing is going to haunt me. Every time I think I've got it fixed, I discover I don't have it fixed. All right. In any event, the cholesterol is a precursor of several things. The steroid hormones, of which there are 5 classes: the mineralocorticoids, the glucocorticoids, the progestogens, the androgens, which are the male sex hormones, and the estrogens, which are the female sex hormones. And cholesterol is the precursor of all those, and you can see those ring structures within there. I didn't say it in class, but again, I'll say it here. Cholesterol is also a precursor of bile acids. Bile acids are very important in digestion. And we'll talk more about those later when we talk about movement of fats in the body. Yeah, the basic structure. Yeah. So this one is another figure that has come to haunt me because I discovered as I was looking at this over the weekend that I had said something very stupid in class. The stupid thing I said in class is that this guy's involved in vision. It's not. And the reason it's particularly stupid is because I used to work in vitamin A. I know this, guys. But it's an example of getting up in front of the class and your brain kind of turns off. The all-trans form is involved in vision, but not the all-trans form of the alcohol. It's the all-trans form of the aldehyde. And you notice the difference. This is an alcohol group. This is an aldehyde group. And this says retinol. It's not retinol. It's retinal. So I've actually fixed the figure, but I haven't posted it yet. And so I'll post it with a note telling everybody, hey, guys, your instructor is an idiot. He said retinol when he meant retinal, and he's got the wrong compound. So the new figure will fix that. But suffice it to say that retinal in the all-trans and the cis form are involved in vision, and they do this kicking thing that I talked about, the bending, unbending, et cetera. And that is what's responsible for helping us to see. These compounds are all made from beta-carotene. It's the primary way we get vitamin A in our diet. And vitamin A, beta-carotene is abundant in green leafy vegetables. Very abundant in green leafy vegetables, which is how you get most of your vitamin A. These are all, of course, fat-soluble vitamins, which is why they're lipids. These lipids include vitamin D, and vitamin D, as I said, comes from cholesterol. You can see part of the cholesterol ring. There's a 6, there's a 6, and there's a 5. The rest of this guy got broken up by virtue of the fact that UV light hits a modified form of cholesterol and causes that bond to break, which gives rise to these compounds. And I said the body is pretty careful in regulating how much active vitamin D that it actually has. But UV light's important in the body's synthesis of vitamin D. Alpha tocopherol is vitamin E. I point out there were 8 different forms of vitamin E, 4 known as tocopherols and 4 known as tocotrienols. They're water insoluble, commonly found in membranes, and they're thought to protect membranes against reactive oxygen species. The last one I talked about is vitamin K, as I pointed out last term. That one's involved in the blood clotting process, and I didn't talk about it anymore here. Mhm. So vitamin D is, so vitamin D is important for calcium and phosphate metabolism. So vitamin D plays a role in determining how much calcium and phosphate you absorb out of your diet and how much you use to make bones. So calcium is really critical inside of cells. Too much calcium, for example, inside of cells can cause DNA to precipitate. That's one problem. Or in the body can cause precipitation and cause problems with things like kidney stones. So the body is really careful about how much calcium it has, and the way it controls that is by controlling how much active vitamin D it has. Okay? You use calcium and phosphate to make bones. So, okay, lipid bilayer was next. There's the lipid bilayer. We see those two tails that we said were nonpolar on the glycerol phospholipids and the sphingolipids, and we see the polar heads on either side. We could think of top, if we wanted to, as outside the cell and down as inside the cell, or we could think of outside the cell here and inside the cell over here, same sort of phenomenon. That's another view of the same thing, and I pointed out this was not a micelle, despite what it looks like right there. I've got to find a replacement figure for that. I haven't done that yet. Okay. Phospholipids, and there's a more close-up view of the lipid bilayer, and pointing out the fact that commonly we see cis bonds inside of here, and those cause bends, and those cause bends, which gives a little bit more chaos and disorder in there. The more disorder, the lower the melting temperature, which means that the more cis bonds you have, the lower the melting temperature. The distribution is uneven. I won't go through that in the interest of time. And the distribution of individual phospholipids in different membranes also vary. You can see nucleus, for example, has a lot of phosphatidylcholine, whereas plasma membrane has relatively little. Okay. I don't think I showed that one in class, but that's a nice depiction of all the different things we see in a membrane. Cholesterol, we see the proteins. You'd say there's an integral membrane protein, there's a peripheral membrane protein. Let's see, what else do we have on there? This shows, for example, that the outside of the cell is where the oligosaccharides are found. You don't see them on the inside of the cell because they play roles in identity, which is important outside the cell and not inside the cell. Okay. Molecular movements, we're getting close to the end. Lateral versus transverse. Lateral movements occurring in the plane of one of the layers of the bilayer. So we can see here, if we stain, as we did here, one layer of the bilayer, we see the thing sort of, this one unfortunately shows black on the bottom as well, but basically we see diffusion in this direction, left and right, not up and down. We don't see the number changing from up to down. And that's because that transverse movement is not favorable at all. Just like movement of other molecules across the lipid bilayer is not favorable, so too is movement of this up to here not favorable. So that transverse movement requires enzymes, and enzymes involved in that are called flipases, as you can see here, flopases here, and scramblases as shown there. Okay. Cholesterol is an important molecule in the membranes. It is fairly soluble in the nonpolar portion of the membrane. And I talked about the melting points of membranes. Membranes should be fluid in order for cells to function properly. So we don't want our membranes to freeze. And so if we look at fish in the ocean that I've talked about before, fish in the ocean, for example, live in a very cold and relatively cold environment, and their membranes have fatty acids that are very unsaturated, which helps them to stay fluid and liquid at a cold temperature, whereas our membranes tend to have more saturated fatty acids compared to fish because we're at a warmer temperature than what they're existing at. The temperature is critical, and the cholesterol influences that temperature, but not the actual value of the temperature, rather the range through which the transition happens. So if we look at this figure, we're comparing solid membranes versus fluid membranes. And we look at those as a function of temperature. So if we start out with solid membranes and we start heating them, we see they go through a phase change to become more fluid as they get warmer, no surprise. And the midpoint of that transition is known as the TM, or the melting temperature, as you may think about it. Cholesterol does not change the TM, as I noted, but rather it widens this range of transition. This appears to be important for membrane function. Yes? Yeah, that's a good question, actually. As best I know, this is not half and half, no. Rather that the character of the membrane itself is changing in that transition phase. So all of the membranes are going through that character change at that temperature. Okay. This is without cholesterol, and this is when you put more cholesterol into it. So the cholesterol widens that range. You see the range. This is shown here basically for no cholesterol, right? So you see that, there's the range. But if you put more cholesterol in that, or put cholesterol in that membrane, now the range gets wider. Okay? Okay. And let's see, fatty acid effects. Again, if you have shorter, more unsaturated, the melting temperature does change. In this case, it gets lower. If you have longer and more saturated, the melting temperature increases. Okay, that finished that topic. And finally, brought us to today. Actually, a little bit of stuff on Friday as well, where we had the lipid bilayer. We see the lipid bilayer is impermeable, particularly to ions. So we see a difference in concentration of ions, a difference in concentration of chemicals. So if we have a charge difference, we have an electro gradient. If we have chemical difference, we have a chemical gradient. And if we combine the two, we have an electrochemical gradient. And that's what we have, as you can see here. Electrochemical gradients are very important for a lot of processes, as we shall see. Concentration we can best understand with osmotic pressure. And osmotic pressure happens when we have a membrane through which material cannot flow. So if I put, for example, a chamber as shown here with water that can flow through here, water will equalize the height on either side. But when I add sugar to it, which can't go through the membrane, now the concentration of sugar is higher over here than over here. Water diffuses through here to try to equalize that pressure. It gets to a point where it's even harder and harder and harder to push. It reaches a point at which it stops. But now because this is higher than this one is, this one's pushing down. There's actually a pressure applied to that. And it's the measurement of that pressure that gives us an estimate of the osmotic pressure. That's where osmotic pressure actually comes from. Osmotic pressure is a driving force for things, as we saw in class today. We also saw it here, where we had a solution that was hypertonic. That is, it had more ions outside the membrane than inside the membrane. Water, which can diffuse through the membrane, comes out. And as it comes out, the cell is left relatively dehydrated. That's a hypertonic solution. An isotonic solution is one that has equal concentrations of ions as what we find in the cells. And so there's no net movement of water, same amount out as in. And a hypotonic is where we have less concentration of ions and compounds outside the cell than inside. When that happens, water diffuses into the cell, and this causes cells to burst. Who was it that asked me about the water not moving across the membrane? Was that you, or was that somebody else? Maybe somebody else. Okay. This is the best example. If you want to talk to a TA and say, hey, water does move across the membrane, that's the best example you can give them. So, okay. Let's see. Membrane proteins. Pretty straightforward. Peripheral, integral, different kind of peripheral. You can see the different forms of that. Associated, and finally anchored. There are two better examples of an integral and an anchored. You saw the thing I showed you today, which is the next figure, actually, that Some people classify anchored as a type of integral. I don't classify it in that sense. But you see the different types that are here. Okay, single crossers with different orientations, multiple crossers, multiple subunit crossers, as you see here. There's the anchored, and there's the anchored with the crosser right there. Terms, a lot of terms here. Uniport, only moving in one direction. Okay. Symport, two things moving in the same direction. Antiport, two things moving in opposite direction. Talked about electroneutral versus electrogenic. There's the electroneutral. We see one ion positive going up, one ion positive going down. They're equalized, so we don't see a change in charge as a result of that. Whereas this, in this case, takes both of them in the same direction, and we see a result, a change in charge. All it takes to be an electrogenic is to have a change in charge by whatever mechanism it actually occurs. Okay, ion channels. Ion channels are basically designed to allow ions to move fairly freely through them. They allow very rapid movement because they don't involve protein shape changes, as some of the systems do. I showed this to indicate an ion channel and the various structural features that it had. And I'll let you just go through that. That's pretty straightforward, I think. Okay, potassium channel was an interesting one in that the potassium channel specifically allowed potassium ions only to pass through it, even though there are smaller ions than potassium ions that occur. And the reason for that is that there's a hydration of ions that happens whenever there's an aqueous environment. So potassium is actually surrounded by waters to which it's ionically attracted. And in order for the potassium to go through this channel, those waters have got to be stripped off. So I pointed out that the unique dimensions, the specific dimensions of the potassium channel, most efficiently allows those waters to be stripped off. And because it's very efficient at stripping those waters off, when they're put back on over here, there's actually energy realized as a result of this process, and that energy realization is what makes the potassium be favorable, whereas the inefficient removal of waters with a different sized ion makes this process be energetically unfavorable, and that's why smaller ones don't make it through, like sodium. All of these different channels have controls for making sure that you can open and close them. Okay? Because if you can't close them, then we've got problems. I talked about different transport protein types, or different types of transport. First of all, passive didn't require a protein. It could just diffuse through the membrane on its own. We could talk about carbon dioxide or oxygen or water or carbon monoxide being passive because it doesn't require anything specific to let them go through, whereas the facilitated, it has a protein that's helping them to go through. So facilitated diffusion happens, but it's still a process of diffusion. Diffusion goes from a high concentration to a low concentration just through the movement of materials. The third process, an active transport, goes against diffusion. It requires energy to go against diffusion, and that energy is required to move at least one molecule against a concentration gradient. Very important point. Yes?
Why is it that steroid-based hormones
Aren't considered what?
just things that can freely diffuse through a membrane?
It turns out that steroid hormones actually can freely diffuse through a membrane.
Right.
They're one of the few exceptions. They can, yeah. So steroid hormones make it through a membrane, and that's only been relatively recently known that they can do that. And they actually hit receptors that are inside of the membrane. Inside of the cell, and they have to diffuse through to get there.
Right.
You know. So what was?
So it's just they're not included in that the big, the five that you mentioned. I'm not sure.
Not included in the. Oh, I see. They're not that common. So if we look at the quantity, I mean, water or carbon dioxide or oxygen, all those are in much greater quantity than the steroid hormones, which are very specific. So that, but you're right, they do go across, but I don't include. I said that the primary ones, and I did when I said that. So, and that's to differentiate the fact that there are some things like the steroid hormones that can make it in. Okay. Okay. good thinking there. Passive diffusion process, you see that. You know diffusion happens, as I said in class. There's the ion channels that allow that to happen. And this is a really good, I'm sorry, this is facilitated diffusion. This is a really good example. I didn't really point it out very well in class, but you can see the shape changes that happens. Here's the protein open. The binding of the ion causes a shape change, and the shape change now allows it to move in. Okay. There's no energy in this case involved in that because the binding of the ion is sufficient to cause the shape change that allows this overall thing to happen. Plus, we're not moving against a concentration gradient. We're moving with the concentration gradient here. Yes, sir?
What causes the ions to unbind?
Yeah, that's a very good question. What caused it to unbind? What do you think would it be?
Just the fact that now the concentration is really low in the cell comparatively, so it wants to fulfill that.
There's two things. One is what you just said right here. Okay. One is that the relative concentration is low, and remember that all these bindings are non-covalent, so we could even bind here and go back upwards, right? So that's one thing. And the other thing is that the shape change actually facilitates a change in the electronic environment that now favors release. Those tiny shape changes are remarkable in terms of what they do. Yes, sir? It's a little bit like that. It's a little bit like that. But T and R, I don't like to use that analogy because I like to save that for enzymes. And this is not a catalysis process. But it is something like that, yes. Okay. Let's see. Did I skip that? Ion channels. I just showed, again, the same thing I showed in the last figure, the change in shape. In this case, an ion channel doesn't have to do that. It's just an opening. Whereas these transporters, which is what you see, transporter proteins, actually do have to change shape. The ion channels are much more rapid in terms of the movements that they allow. Active transport involves many of the same things. However, energy is involved, in this case ATP. That's involved both in the shape change and in the movement of the molecules from low concentration to high, which is what we have to have in order for active transport to occur. Sodium potassium ATPase, I pointed out, was the most important transporter inside of cells, moving 3 sodiums out and 2 potassiums in. I got to change that figure, 2 potassiums in. And that makes it an electrogenic transporter. It also makes it an antiport because we've got out and we've got in. Okay. It is a pump, yes, exactly what I would call it. I call it the sodium-potassium pump. That'd also be fine. Yep, certainly. Yep. It's called an ATPase because it breaks ATP down in the process, which is why they call it that. But it would be perfectly acceptable to call that a pump. I didn't talk about the steps in this process, and to be honest, I don't think the steps in the process are particularly relevant. But the important thing here is that the cell is recalibrating osmotic pressure. It's doing some very cool tricks to maintain osmotic pressure so the cell doesn't burst. Very important thing. Another pump I talked about that didn't require ATP was the sodium glucose pump. It's driven by the high concentration of sodium ions outside that want to come in. And when they come in through this pump that bring in the glucose with it. The glucose is moved from a low concentration to a higher concentration, which is why this is an active transport system. And the energy comes from the sodium gradient. What else can I say? That's pretty much it. The proton lactose pump, that is the lactose primase, uses protons as the driving force, and I pointed out that the protons bind here, and it's the binding of the protons here along with the lactose that causes the shape change that actually moves this into the cell. And again, no ATP involved in this process. But the proton gradient is the important energy source. The proton gradient is shown a little bit better here. A proton pump kicking things out, causing a high concentration of protons out, which then drives the movement of the lactose in. And nerve transmission, the last thing to talk about. So nerve transmission is possible because of gated ion channels. Those gates are kept in a closed state until a signal is received, and that signal can be, if it's a touch, a nerve cell that responds to the physical nature of what is involved in touching something. It could be an auditory thing, as in your nerves. It could be taste. It could be a variety of things. But suffice it to say that the stimulus causes the channel to open. And when we do nerve transmission, the channel that opens first is the sodium. Sodium channel opens. Sodium ions flood in. We see a change in concentration of sodium. We see a change in the charge. So we've generated an electrical signal at that point. And electrical signals are critical for stimulating other ion channels to be affected in nerve cells. So the potassium channel becomes affected. The potassium channel then opens. Potassium starts flowing out, neutralizes the charge. We see a voltage change that happens as a result of that. That movement of sodium in and potassium out is communicated further down the cell, so that this voltage basically is moving down the cell, and then the next sodiums and potassium gates open. It goes all the way down the length of the nerve cell until it gets to the very end, at which point neurotransmitters are released. The neurotransmitter release goes across the synapse to another nerve cell, which starts the same process over and over, and that travels all the way ultimately up to your brain. A remarkable process. Yes? I'm sorry? How is it not going backwards? Okay. So, why is this not going from right to left, basically, is what you're saying, right? Okay. So, if I get an influx of sodium ions, I'm going to have a higher concentration of sodium ions here, right? What's going to happen here? Diffusion is going to drive it to the right. That's what's going to happen. So that movement is automatically chemically driven as a result of the concentrations that are there. So that's why we see the voltage changes that happen. Moving charges change voltage. So there's literally, it's called an action potential, but there's literally a movement of voltage all the way down there. And of course, the beautiful thing about this is that the movement of the voltage favors creation of more voltage, because it favors the opening of the next ones, which causes more sodium ions to flow in, and it just goes all the way down. And that's the fact that we have an ion channel makes it rapid, but this opening makes it very efficient. If we look at the actual signal that happens with this, okay, it is propagated along the nerve cell as efficiently as electrical signals go down lines. That's how efficient it is. It's remarkable. It's not as fast as an electrical signal, because that can happen at the speed of light. Okay, and this isn't happening at the speed of light. It's happening at the speed of diffusion, but that involves a few milliseconds. It doesn't take very long to make that happen. Remarkable. Okay, so we're at the end of the nerve cell. The end of the nerve cell is where the neurotransmitters will be released. They go from here over to the next nerve cell and cause the same process to happen over there. And with that, that's as far as I got today. I didn't get to talk about neurotoxins. I'll mention them briefly next time. Maybe to finish it off here, I'll just go ahead and mention them here as well. Neurotoxins are compounds that will affect some of these channels. Okay? If they affect and they interfere with the transmission of nerve signals, they can cause some real problems. They're toxic in the sense that they block the channel and keep the channel from opening and closing as it should, and consequently, basically it can kill the nerve cell. But tetrodotoxin is one such toxin. It's the active ingredient of things in pufferfish that kill people. So you've heard of pufferfish, which is a delicacy in Japan. And if you don't properly remove the toxic part of the pufferfish, you can die from the toxin that you get from that, and that toxin is known as tetrodotoxin. Another toxin that comes from the sea is this guy right here, saxitoxin. Comes from shellfish that get it from algal blooms. So they monitor algal blooms in the ocean pretty carefully. When you hear about occasionally they get a shellfish fishery or whatever we call it closed down, it's because that they've detected something, this or other things, there's others as well, that are being produced and being consumed by these shellfish that they're catching. And if you eat shellfish that has that, you're going to have some nasty effects as a consequence. That was an hour and a half. That was a lot of talking. I hope that wasn't too fast, but I was trying to get through everything so I could explain. Are there any questions or anything I can answer for you guys? Clear as mud? Yes? If you consume too much sodium or potassium? Well, yes, but particularly potassium, actually. Potassium, you can get too much potassium and cause some problems. The main problems that you see, it's kind of hard to do, but you have to more likely inject it than anything else. But the main problems that you see actually relate to sodium because there are effects of sodium on blood pressure, for example, and that's more commonly the things that you would see. Yeah. When they do lethal injections of people, where they give them these various drugs and so forth, part of the lethal injection is commonly a big chunk of potassium, which basically stops the heart. So, yeah. But you would probably, I'm not an expert in this, but you probably would have to inject that level to get it in the bloodstream. I don't think the body probably would absorb that amount. I don't know that, but I would guess that. All right, guys. I don't know if you've eaten dinner or not. I haven't. My stomach is grumbling, so let's go get some dinner and study hard, okay?